To the last speaker of the session, and that is José Javier Lucas, and he's going from the CBM in Madrid, and he's member of CIBERNED, and his talk is focused on altered RNA processing in Huntington's disease.
So, good morning, everyone. I would like to thank Fundación CIEN and CIBERNED for giving me the opportunity to present today results from my laboratory, where we show that there is a global alteration of RNA processing in Huntington's disease. And I hope I will be able to convince you that such global alteration in RNA processing significantly contributes to the pathogenesis of Huntington's disease and also offers new opportunities for therapeutic intervention for this devastating disease for which no modifying treatment is available nowadays. So let me start by giving a brief introduction of what we call RNA processing. This is a series of modifications that RNA molecules experience as soon as they get transcribed in the nucleus and until they are in the cytoplasm for presentation to the ribosome for protein translation. And these modifications are carried out by a plethora of RNA-binding proteins that are depicted here as these color shapes. And we have evidence and to explore why, if these different modifications occur differently in Huntington's disease. And I will show you that one of these alterations that it is splicing, that, as you know, is the removal of non-coding regions of the transcripts that are the introns to leave only the coding ones, the exons, is what is called splicing. But splicing not always happens in the same way for a given transcript, and that originates what is called alternative splicing. That means that not always all the coding sequences are included, or they are included in different ways. And by doing this, the cell gets a bigger proteome repertoire from a single genome. This phenomenon of alternative splicing increases along evolution, and particularly in the brain. And the other RNA processing that we will show that is altered in Huntington's disease is regulated polyadenylation. The poly-A tail is added by default in the nucleus to confer stability to the messenger RNA, but then this can be regulated also in the cytoplasm, as I will explain later. So let me give you also, probably in this auditorium you don't need much introduction about Huntington's disease, but just to briefly remind you that this is an strictly inherited neurodegenerative disease, that it is caused by a fully penetrant autosomal dominant mutation, and thus had allowed the generation of bona fide animal models. The neuropathology affects mainly the basal ganglia. Particularly, there is an atrophy of the striatum and also a shrinkage of the cortex, and that basal ganglia pathology explains the symptoms that are mainly motor, but also psychiatric and eventually also dementia. And the mutation is an expansion of a CAG repeat in the coding region of the Huntington gene. And when the repeat is above 40, it is when the disease will take place, and the longer the repeat, the more severe the symptoms and the earlier the age of onset. This CAG repeat is, as I said, is in the coding region and encodes a polyglutamine sequence that is very well documented, that is toxic and also confers a propensity to self-aggregate to the mutant Huntingtin that accumulates in inclusion bodies, and also causes new protein-protein interactions. But apart from this very well-established toxicity of polyglutamine, there is also a very well-documented toxicity of the CAG repeat in the mRNA, as I will also show in detail later. And in our laboratory, we have recently established the signatures of missplicing and mispolyadenylation in the striatum of Huntington's disease patients, and this has allowed us to identify potential new biomarkers of disease progression and also new targets for therapeutic intervention. And regarding splicing, the interest in alternative splicing in Huntington's disease started approximately like a decade ago. And the first report was this study by Gillian Bates in London, where she found that the splicing of the Huntington gene itself is altered in the disease, probably because of this CAG repeat in the first exon. There is an incomplete removal of the first intron, and these few molecules that contain the intron, because there is a cryptic polyadenylation site in this intron, can act as a mini gene that produce a very small but very toxic version of mutant Huntington that is exon 1 and terminal Huntington that is very prone to aggregate and also to interact with other proteins. And slightly afterwards, we reported an alteration of splicing isoforms of tau that we have already heard yesterday and today. You know, this is the protein that aggregates intracellularly in Alzheimer's disease, but also is altered in other diseases called tauopathies. And particularly, we found alteration in exon 10 of tau that encodes one of the tubulin binding domains of tau. So as a consequence of this imbalance in isoform splicing isoforms, we get an excess of 4 repeat tau that has a higher affinity for tubulin and also to self-aggregate, and mutations affecting this splicing of exon 10 are responsible for some rare forms of frontotemporal dementia linked to chromosome 17. And we were able to ascribe that alteration in tau splicing in Huntington's disease to an alteration on the splicing factor SRSF6 that had already been reported as a splicing factor able to modulate exon 10 splicing, and we found it accumulated in the inclusion bodies in the brains of Huntington disease patients and mouse models. And that was quite some time ago, but more recently in the laboratory, a former PhD student, Ivo Hernandez, that is now doing a postdoc in Oscar's lab, Ivo Hernandez performed a study on an interactor of SRSF6, that is this other splicing factor called SREG1, that they were known to physically interact, and they were postulated to modulate their activity. And this was very interesting for us because one of the known or postulated targets of SREG1 was TAV1, and this was very interesting because mutations in TAV1, particularly a retrotransposon insertion that results in decreased TAV1 expression, is responsible for a very rare disease, genetic disease endemic of the Philippines that is called X-linked dystonia Parkinsonism, or Lubac disease of Panay dystonia, that is characterized by a marked atrophy of the striatum, same as Huntington's disease. And summarizing in a single slide a lot of work by Ivo, he was able to demonstrate that by decreasing SRSF6 suffices to induce a decrease of SREG1, which in turn leads to missplicing and decreased levels of TAV1, therefore showing a molecular mechanism convergence between Huntington's disease and XDP. As I mentioned, XDP is caused precisely by a decrease of TAV1 expression. And I haven't mentioned that TAV1 is the bigger and scaffolding protein of the huge complex that is a basic transcription factor, 2F2D, that is involved in the pre-initiation complex of transcription. I don't have the time to go into those mechanistic details today, but I would like just to mention that we have created mice with CRISPR modification of TAV1 to induce to force the expression of one of the splicing isoforms, and this results in a very interesting neurological phenotype that we believe explains part of the disease in Huntington's disease, but probably we will need the help of some of you to continue the characterization of these mice and the molecular mechanism. And so at this point, we had already 3 genes linked to neurodegeneration, Huntington itself, tau, and TAF1, that are misspliced in Huntington's disease. And we realized that it was no sense on keeping analyzing them one by one. So it was mandatory to establish what is the global signature of missplicing in the striatum of Huntington's disease patients. And for that, Ainara Elorza in the laboratory had to develop a new approach that we call human-mouse intersect RNA-seq, because interrogating this missplicing signature is not trivial for this reason, that is that you have this huge atrophy of the striatum in which you have a massive neuronal loss, but you also have gliosis and inflammation, and alternative splicing is cell type specific and also affected by inflammation. So if you just simply compare the splicing status here and here, probably most of the differences you are going to pick are going to be consequence of the difference in the cellular composition and the inflammation. So to overcome this, Ainara took advantage of this mouse model that has a quite robust phenotype, but that if you sacrifice the mice at an early stage when they already have motor symptoms, they still do not have neuronal loss nor gliosis. So by performing deep RNA-seq of these 2 types of samples, the human and the mouse samples, and by obtaining this common signature of misspliced genes in the striatum of the patients and of the Huntington mice, we reason that probably this signature is going to be the result of the toxicity of the mutation rather than the adaptations and the compensatory changes in the tissue. So then we follow this workflow with the data mining, screening with basis of Mendelian diseases, and to identify the common event in both species, RT-PCR validation and Western blot analysis. And this allowed us to identify 6 genes that are linked to 6 neurodegenerative or neuromuscular diseases, and whose missplicing in the striatum of Huntington disease correlates with decreased protein levels of these genes, and therefore each of them becomes a new target potentially for therapeutic intervention, same as TAF1 or tau that we have found before. Also, as part of this study, Ainara performed a bioinformatic analysis of the consensus motif for the different splicing factors in the flanking regions surrounding those exons that are coincidentally misspliced in both species, and that way she was able to find 5 candidate splicing factors that whose protein levels were also decreased both in the human tissue and in mouse models, and that might significantly contribute to this pathogenic mechanism. And we are very proud that Jising, who is the one who developed the algorithm for RNA splicing analysis, RMATs, that we all use, commented this study recently in teens, underscoring that this approach can be used to any other neurodegenerative disease for which bona fide animal models exist, like for instance, familial ALS. And nowadays, we are exploring which of these splicing factors might be contributing the most to the missplicing signature, and we are currently focusing on RBFOX because we found that RBFOX targets are predominant within the missplicing signature, and this led Ainara Elorza and David Lozano in the lab to generate mice overexpressing in the brain RBFOX1 at different levels, and they found that the mild overexpression of RBFOX1 attenuates the motor phenotypes and also the neuropathology, and they are characterizing the specific correction of the missplicing events. And hopefully we will report that soon. And so that's all for the part of splicing. And just summarizing, apart from tau and TAF1, we have been able to identify these 6 additional misspliced genes that might be important effectors of the phenotype, and also identify an additional splicing factor on top of SRSF6 and SREC1 that might be contributing to the missplicing signature. And regarding the other RNA processing modification, regulated polyadenylation, the reason why we thought this might be altered in Huntington's disease comes from a series of elegant experiments from the group of Nancy Bonini. Actually, they were working on a different disease, that is spinocerebellar ataxia 3, that is due to mutation in ataxin 3, but it's the same kind of mutation. It's an expansion of a CAG repeat encoding a polyglutamine sequence. And there are good fly models, Drosophila models of neurodegeneration with ataxin 3. And this experiment demonstrated that the CAG expanded RNA is toxic, because you can see here how expressing this transgene with the expanded CAG giving rise to the corresponding expanded polyglutamine in ataxin 3 induces a profound degeneration of the eye in the Drosophila. However, if you produce the same protein, the same ataxin protein with the same polyglutamine length, but encoded by an RNA that is not a CAG repeat, but intercalating CAG and CAA codons that they encode polyglutamine, then you don't get so much toxicity. And then afterwards, they perform a screening of modifiers of genes that are able to attenuate the toxicity of either the pure protein toxicity or the pure RNA toxicity. And there were only 5 genes able to counteract both toxicities. 2 of them were chaperones, no surprise, because of the protein aggregation phenotype that I mentioned before. There is one lipid-binding protein that maybe not so much surprise after yesterday's presentation of Tom Sudo showing the important role of cholesterol metabolism in synaptic dysfunction. And the other 2 were RNA-binding proteins. One was a splicing factor, muscleblind, and the other one, ORP2, is a protein involved in regulated polyadenylation. ORP2 is the Drosophila ortholog of CPB2, 3, and 4. It's a family of RNA-binding proteins with an additional member in mammals, CPB1. And what these proteins do is that they bind this EU-rich sequence called cytoplasmic polyadenylation element that is located near the poly-A signal in the 3' UTR untranslated region of many transcripts. Approximately 40% of the transcripts have these bioinformatic requirements to be regulated by these proteins. And what these CPB proteins do is that they bind to the target RNA and nucleate the formation of a protein complex that catalyzes the deadenylation, the loss of the poly-A tail, and keeps the messengers in a dormant state that is not translated. However, when the right signal comes, maybe through a translational modifications like a phosphorylation or ubiquitination, then there is a change in this complex, and now the poly-A tail is added back again, and the messenger is presented to the ribosome for efficient translation. So this way, these proteins confer temporal and local regulation of their transcript RNAs. And for this reason, we decided to explore whether they were altered in Huntington's disease. And what Sara Picot and Alberto Parras found in the lab is that there is an increase of CPB1 and a decrease of CPB4 in the striatum Huntington disease patients. The same is found in the mouse models. And this led us to analyze the status, or the global status of polyadenylation in the transcriptome by performing a polyuchromatography that allows you to generate fractions of the RNA enriching long poly-A tail transcripts and fractions enriching short poly-A tail transcripts. And by comparing them in the wild type and in the Huntington mice by gene chip analysis, we were able to find that 9% roughly of the transcript has elongated poly-A-tail, while 9% has a shortened poly-A-tail. And the later were pretty much interesting because those genes with the most marked deadenylation that is supposed to correlate to lower expression of the protein with lower translation, among the top deadenylated transcripts, we found 2 genes related to striatal atrophy that had never been related to Huntington's disease. One is kinectin, one that I don't have the time to present today, and the other one that was very interesting is SLC19A3. It's a solute carrier protein. That was very interesting because, again, mutations in this gene produce a basal ganglia disease that is very interesting because it reverts with a combination of vitamins. The disease is called thiamine, biotin and thiamine responsive basal ganglia disease, BTBGD, and it responds to high doses of thiamine or vitamin B1 because SLC19A3 encodes one of the transporters of thiamine, THT-TR2, and those in high doses of thiamine compensate the decreased transport, and also in high doses of biotin are known to change transcription, including increasing transcription of this messenger RNA. And therefore, this is BTBGD is a form of Leigh disease in which the deficit of thiamine, that is this transporter is the predominant one in the brain endothelium, results in decreased thiamine and more precisely thiamine pyrophosphate, that is the cofactor of many enzymes, some of them in the mitochondria that are responsible for the symptoms of this disease. So we reason that maybe the deadenylation of this gene in Huntington's disease leads to decreased protein levels that would result in a similar situation as what happens in the patients with mutations in the gene. And to test this, we explored the levels of the transporter in the striatum of Huntington's disease patient, and it is actually decreased not only in the striatum, also in the cortex. And in good agreement with the Human Protein Atlas, the decrease takes place in the endothelium, but also in the neurons, as humans express this transporter also in neurons. And next, we analyzed the levels of thiamine in the cerebrospinal fluid. As you can see here, the prevailing form of thiamine in CSF, that is the monophosphate form, is significantly decreased. And more importantly, in the brain parenchyma, there is a decrease of the TPP, the cofactor form of thiamine, and this allowed us to conclude that, in fact, Huntington's disease is a brain thiamine deficiency similar to what happens in BTBGD, that they have this decrease in CSF thiamine despite normal blood levels of thiamine. And therefore, our working model is that the mutation in CAG leads to this alteration in the CPVs that lead to deadenylation of the transcript that ends up with lower levels of the transporter and lower levels of TPP. And this offers a clear opportunity for therapeutic intervention, and that's what we tested preclinically in the mice. And we found that the treatment attenuates the motor phenotype and also the neuropathology, and this has originated this study that is a collaboration of 4 CIBERNED groups in which we propose a TMP, the monophosphate thiamine, as a potential biomarker of disease progression in Huntington's disease. And that's something we are exploring in collaboration with Jaime Kulisevsky and his team at San Pao Hospital. And we have recently requested for funds to do this in the collaborative project called of CIBERNED. And also, this has originated a clinical trial in collaboration with Pablo Miras in the Hospital Virgen del Rocío in Seville that is intended to test safety and tolerability of these high doses of the two vitamins that are, although they are used in BT-VGD, they have never been used in HD patients. It's only just 4 patients. It's an open-label study, but hopefully the secondary biomarkers might give us some clues that it might be worth trying in a bigger clinical trial to explore efficacy, but that would be the need in a second round. Last month, the patient advocate portal of Huntington's disease, HDPAS, publicized this clinical trial, and we are very excited about this. And just as a summary for the therapeutic implications of the work I've presented today, I would like to remind you that we have found in the past an alteration of tau splicing. More recently, we found an alteration of TAP1, and also all these candidate effector genes that are amenable for correction through a technology that is alternative splicing correcting oligos, similar to the silencing oligos that are so trendy as a way to cure Huntington's disease. But unfortunately, for the moment, the delivery, the availability to the striatal tissue probably is not efficient enough to download the levels of Huntington enough, but maybe in combination with these alternative splicing correcting oligos that are already in the clinic, as we heard yesterday, for spinal muscular atrophy with nusinersen, and there are many clinical trials for ALS with similar drugs. Maybe in combination they might help, but they are not trivial, so it is important to keep investigating possible new easily druggable targets, and in that sense, TAP1 might be one of them, because we have evidence that the missplicing is decreasing the protein levels because there is a new protease target sequence that can be counteracted with inhibitors of the proteases. That's something we are testing now. And definitely very easy to implement is the vitamin therapy that is already reaching clinical trials. And also we have found a potential biomarker. And that's all I wanted to present today, just to thank the fantastic team that carried out the work. I think I mentioned already the names of the people involved and our excellent network of collaborators. My apologies if I'm forgetting anyone. And also to thank the funding bodies. And very important also the brain banks and the families for providing the samples for the studies. And thank you so much.
Thank you, José. We have time for one quick question. And we have to run for it.
Okay, José, that was very nice. So what about the order of events? So you see problems in the general transcription factor, in splicing, polydenylation. So what is first, or does it co-occur? Can you see the primary problem?
Well, we are not able to establish a chronology of all these. These are probably multiple events happening simultaneously because of different molecular mechanisms. I didn't have the time to get into why all this is happening. So some of the splicing factors get altered because they get sequestered into the polyglutamine inclusion. Others, probably we don't have data on that, but other groups have. They are sequestered into the hairpins that the CAG repeats cause. So, and the transcriptional alteration, well, once all this is happening, everything is happening together. Probably each of these will have a modest effect, but altogether, it's what it's probably once with aging, you have your homeostasis, your fitness is not so powerful along the life. Then is when all these insults that are modest insults, but happening all of them together at the same time, you reach a point in which the symptoms appear when the patient is 45 or 50 years old. But probably they are all happening in parallel with a mild toxicities that they are summing up.
Before we close the session, I want to remind you that coffee is served in the claustro, in the building, in the continuation of this building, in the hospederia. And we remind you that we have to be back here sharp at 11:30. And I want to thank the speakers for the beautiful talks, and thank you all.